Hi students, welcome to Vivid Botany. Here we have a question about gel electrophoresis. We know that during the digestion of the DNA fragments by restriction endonuclease, the digested DNA fragments are separated by the gel electrophoresis. So, in this gel electrophoresis, what is being used as gel? Gel electrophoresis. In this process, what is being used as gel? We know that agarose is used as gel. Agarose is a natural polymer. It is obtained from Gelidium and Gracilaria that are of red alga. So agarose will provide sieving effect for the movement of DNA particles, and DNA being acidic, being negative in charge, it will migrate towards the positive electrode, that is anode. DNA fragments which are larger in size will migrate slowly. DNA fragments which are smaller in size will migrate fastly. More digested particles are smaller in size, less digested particles are larger in size. So that is the case of electrophoresis. It is discovered by Dr. E.M. Sutherland. So it is also called as Sutherland blotting technique. It's also called Sutherland blotting technique. So in this Sutherland blotting technique, DNA fragments will migrate towards the anode, that is positive electrode. So we have here another term that is called acetocarmine. Acetocarmine. Another I'll get you is acetoarsin. Acetoarsin. This acetocarmine and acetoarsin both are basic dyes. So these basic dyes are used to stain the chromosomes. We know chromosomes. Chromo means colored, some means body. So colored bodies. How they get colored? Because they are stained by the basic dyes acetocarmine and acetoarsin. When they are stained, they appear red in color. So red colored bodies which are elongated rod-like structures are called chromosomes. They are discovered by Valdair. They are made up of the chromatin reticulum. Earlier it was in prophase, and after the spiralization, condensation, they convert into chromosomes. So this chromatin reticulum, when it is stained by acetocarmine and acetoarsin, it will give two types of colorations. The word chromatin is coined by Fleming. It is the reticulum of nucleoprotein fibers. Reticulum means network like. Nucleo means nucleic acid. Protein means histone proteins that we know, H2A, H2B, H3, H4, H1, all that. So this chromatin, during its stained, that is after staining, it will form two types of colorations. One is heterochromatin, and the other is euchromatin. Heterochromatin means it is densely stained. Euchromatin means it is light stained. So what is their significance? Densely stained means it is having the non-functional codons, that is, they are having inactive genes. This heterochromatin contains inactive genes, whereas the euchromatin contains active genes. And one more interesting feature is that heterochromatin is tightly coiled, that is inseparable. Euchromatin is loosely coiled, that is separable. So having the two types of chromatin after staining is called heteropyknosis. This feature is shown by the chromatin reticulum. So that is about acetocarmine and acetoarsin. Acetocaramine and acetoarsin both are used to dye the chromosomes, that is DNA material. Now let's come to the second option, that is Ethidium bromide. What is this Ethidium bromide? Ethidium bromide is also a dye. It is also a dye. In fact, it is called as cellular marker, a non-radioactive cellular marker for identifying and visualizing nucleic acid bands in electrophoresis. It is used as non-radioactive marker to identify and visualize nucleic acids, to identify and visualize nucleic acids in the form of bands, in the form of bands in process called as electrophoresis. Electrophoresis, since we make use of agar gel that provides sieving effect, we call it as gel electrophoresis. So this is a crystalline non-volatile powder that is soluble in water. So this Ethidium bromide fluoresces a red-orange color under UV light. This eth bromide fluoresces means it provides the fluorescent a red-orange color under UV light, that is ultraviolet light. We know UV light causes mutations also. UV light causes cancer also. We know ozone depletion is making UV light to directly fall on the earth. So what is the function of this UV light? This UV light that excites the electron within aromatic ring of Ethidium bromide. UV light excites. We know that excitation means, that is, when we provide energy to an atom, the photoelectric effect takes place. That is, electron present in the valence shell or the outer orbital will eject out of that atom. It is called cationization, ionization, oxidation. So that is called photoelectric effect. So this UV light will cause excitation of an electron, within aromatic ring of Ethidium bromide, aromatic ring of Ethidium bromide. So when they return to their original state, original ground state, when that electron return to their original ground state, the light is emitted. The light is here emitted. So two factors are necessary for making DNA fragments to be visible. One is Ethidium bromide, another is UV radiation. If you see the answers options here, in the first option he gave acetocaramine in blue, bright blue light. This is wrong, because acetocaramine is used to dye, stain that chromosomes and chromatin reticulum. Second option, Ethidium bromide in UV radiation. This is very much correct. Acetocaramine in UV radiation, it's also wrong. Ethidium bromide in IR radiation, this is also wrong. So it is a jumbling of the words that are options given in this question. The words are jumbled here. That is, Ethidium Bromide once with infrared rays, acetocaramine once with bright blue light, acetocaramine once with UV radiation. So the question is a straightforward question as far as my view is concerned. The only thing is that students should have an excellent comprehension of the concept of the agarose electrophoresis of the southern blotting technique and how the DNA fragments appear. They appear orange red color bands, and which radiation, that is UV radiation, addition of what, that is ethidium bromide. So this type of concept should be very clearer, concrete for the student. Then only he can pick up the right option among the all. So I think we are going on right path. The time is nearing for our the most awaited competitive exam NEET 2022. So we will be doing all the questions one after the other. I will be trying to clearing as many concepts as I can through my video channel, that is Vivid Botany, and I would like to have your responses in the channel, that is YouTube channel Vivid Botany. Make a comment and drop your responses, and even you can ask any doubts also in the YouTube channel, so that we can have our next session discussing about their doubts also. So please not to forget to make your comment and to drop your response in the channel, and please subscribe my channel, that is Vivid Botany, which is mainly focuses on the botany competitive exams like NEET UG. So we are not neither entertaining nor passing time, just we are meant for only those students who are very serious towards the exam. So hope we are not wasting my time and also your time also. So do subscribe my channel, that is Vivid Botany, and drop your comments. Thank you.